Okay, so it's a pleasure to introduce James DiFrisco, who is our speaker this week and has actually spent the last week with us. And so you are a postdoc position you have in Belgium. Philosophy is a philosopher. And, today, I think that he will just deconstruct the core of the Wagnerian view of evolution. And we are all looking forward to this. Of course. Always make it so easy on themselves.
Thank you. I won't exactly do that, but do something else. So thanks for inviting me to present my stuff, and spending last week here has been really stimulating. So thanks for that. So my talk today is about the notion of characters or traits. I'll be using these terms interchangeably. I won't enforce a rigid distinction. There are some distinctions, and I'm open, but I won't enforce anything. So this is more about an exploratory talk, not really a finished paper or anything. So I'll be happy if I can give you at least one interesting idea or initiate an interesting discussion. Also, feel free to interrupt me throughout. So here's what I'm going to cover. First, some setup. What are the roles for the notion of characters in biological reasoning? Then I'll discuss a little bit of methodology. So what kind of problem is this for conceptual biology? I'll compare it to the problem of biological individuality, organism identity. I'll talk a little bit about developmental-based notions of characters and evolutionary characters, and end with some more speculative stuff about what I think is some work for a theory of characters, using theory in a very loose sense. So let's begin with some basic distinctions. Characters can be many things. What are characters? They can be body parts, like the vertebral column, insect segments, cell type. Characters can be properties, so color, shape, size, allometry. More specifically, they can be variables that take on mutually exclusive values, like height, mass, generation time, age at maturity, and so on. Characters, you can also have activities or organism behaviors or even functions treated as characters. I won't really talk much about this today. Some examples would be growth, gastrulation, mating displays, or so on. Alan Love and Ingo Briggen have done some interesting work on this. I won't really talk about it. The other distinction to note is that characters form hierarchies of specification. So, for example, you have the vertebrate forelimb, then avian forelimb, raven forelimb. For any given character at one level of taxonomic specification, the characters at the next lower level are states of that character, or character states. So you have this character versus character state distinction. So with this general background, we can look at some roles, the notion of character in biological reasoning. This is a pretty loose list. It's not exhaustive and subject to revision, but this just gives you an idea of some of like the diversity and wide extent of usages and roles. So one is that characters are used or should be used in measuring organismic complexity. Complexity can be measured in terms of number of cell types or number of character types. Complexity figures into many generalizations in evolutionary theories, such as concerning the cost of complexity, or the idea that complexity generally increases in evolution. So if those ideas are to make sense, there must be some notion of character in the background, some way of counting characters, right? Similarly with measuring pleiotropy, so a gene is pleiotropic if it influences more than one trait. Well, how do we know that the traits are really more than one? How do we know that they're really distinct? If you want to measure pleiotropy, again, for purposes of theorizing about something like cost of pleiotropy, we should have some kind of working criteria of identity and distinctness for characters. Characters are also obviously used in the context of distinguishing phylogenetic groups. Characters distinguish taxa. Then you also have a role for characters in identifying the bearers of fitness or the contributors to fitness. So expressed differently, this is about understanding the true causes of a selection process. And this is related to the challenge of identifying causally, the true causes, the traits that are causally real rather than epiphenomenal, that are really undergoing selection. Characters have a role in just simply describing morphological change and tracking change in populations. So characters are descriptive and functional building blocks of morphological and other kinds of evolution. Also, you have a role in identifying evolutionary novelties. And I think this is interestingly similar to a problem in the context of biological individuality of identifying organisms, of basically distinguishing between what is reproduction of a new individual and what is a continuation of the growth and development of the same individual, of the parent, right? And those kinds of questions are fairly clear in vertebrates, but not in other kinds of organisms, such as like marine invertebrates with really complex life cycles are a lot of fascinating examples. So it's similar to that question. The notion of characters also comes up in the context of weighting rival phylogenetic hypotheses. So the number of characters or number of synapomorphies establishes the weighting of rival phylogenetic hypotheses. For example, if one proposed taxonomic grouping has 10 shared characters and a rival grouping has 3, then the hypothesis that the first one is monophyletic is more parsimonious. And obviously in this context, the way you count characters influences the number for each group and thus the weighting of the hypothesis. And these methods sometimes assume that different characters constitute independent pieces of evidence for phylogenies. But then you can ask questions like, well, does that presuppose an implausibly strong degree of modularity in characters? And how do we know when characters are really independent pieces of evidence? So I'll return to that with a specific example. Another general role is just in measuring rates of evolution. You have to establish scales and comparable units. So I talked about this with Thomas the other day in quantitative genetics. Apparently there's a problem with establishing comparable scales. So if we're saying if one trait evolves on a ratio scale and another trait evolves on an interval scale, can we say which trait has a higher rate of evolution or which trait is more evolvable? It's not clear. Generally, also, characters are involved in assigning differential evolvabilities. So here's a quote from the paper from the reading group yesterday. He says, the first difficulty faced in studying G is finding a non-arbitrary way of choosing the traits to be used to construct the variance-covariance matrices themselves. And this seemed to be a point that people generally agreed on, right? So how can you make this choice non-arbitrary? I don't think that the methodological advice, measure everything, is very good or very practical. So you need to find a way of sort of reducing the dimensionality of the phenotype and figuring out what you should be measuring. And then there's just a very general role in establishing comparable units of phenotype. So I think that the character concept really becomes important in the context of comparative biology. So for example, studies of convergent evolution, you need to establish relevantly similar traits to compare. So again, not an exhaustive list, but you get an idea of the diversity of roles. So on this basis, we can, to a first approximation, state what the problem is, the problem of character individuation or individuality. I'm going to say the problem is to provide criteria that successfully identify characters that fulfill those roles. So for example, which characters are good for distinguishing phylogenetic groups? Ideally, these would be apomorphies that arose once, not homoplasies that arise repeatedly. Right? Which characters are good for comparative studies of convergent evolution? Well, these should be phylogenetically independent characters that have ideally arisen more than once in the course of evolution from different starting points. So these are really quite different demands on the notion. Another example, what kinds of characters should be used when identifying the bearers of fitness or contributors to fitness? Well, here we probably want a functional character concept rather than one based on history or phylogenetic origin. Homologs can, of course, acquire different functions and different contributions to fitness in specific lineages. So you can already start to see how the concept of characters is pulled in different directions by different roles. So it seems like initially that a single unique definition of characters is quite unlikely to be found. So now I want to compare it to the organism individuality question. Posed in this way, I think it's fundamentally similar. So it's a problem of identifying the roles in biological reasoning where differences in your conception of individuality make a difference for your explanations, predictions, or understanding of natural processes. So the problem is to then find those criteria of individuation that allow you to satisfy the roles successfully. And this is the line of thought that I pursued in this paper about organism individuality. So given this set of roles, the exact content is not important. I argued that the available criteria cluster into 4 basic kinds of individuals: evolutionary, physiological, ecological, and developmental. So I want to pursue the same line of thought with the problem of character individuality, and my intuition is that there's going to be a very similar kind of clustering of criteria when it comes to characters. So as a preview, I'll distinguish between a few kinds of characters. We can sort the criteria into these broad categories. Developmental notion, evolutionary, and I'd say this splits into a functional evolutionary and a historical evolutionary notions, an ecological notion, and then perhaps there are more. So in this approach, the term character by itself is not meaningful without further specification of the kind of character that we're interested in. Often this is implicit in the context, but often it's not. So back to organism individuality, people sometimes ask questions like, is a worker bee an individual? Is an aspen forest an individual or organism? I think we need to specify what kind of individual we're talking about before that question is really meaningful. So I argued that a worker bee is a developmental individual and a physiological individual, but not an evolutionary individual. A colony is the evolutionary individual. This is an obligately eusocial species. So because these different kinds of individual can be dissociated, we shouldn't bundle them all together under the vague heading of organism or individual. And I think the same is true of characters. So we shouldn't ask questions, I think, like, is the human chin a character? Is age a character? Can a developmental process be a character? Instead, we should ask, is the human chin an evolutionary character? Is or is it a developmental character, and so on. So another ilk on this same perspective. In the editorial introduction to this book, Gunther Wagner gives a kind of first approximation definition. A biological character can be thought of as a part of an organism that exhibits causal coherence to have a well-defined identity and that plays a causal role in some biological process. So the thing is, there are different processes that characters can play causal roles in. So the same character might be a unit of an evolutionary process, but not necessarily a developmental process. So let me make this stuff more specific. Let's look at an example. Color patterns on the wings of Lepidoptera form a system of characters known as the symmetry system. These color patterns are produced by local events of pigment synthesis that result from enzymatic pathways interacting with the vein compartments on the wing. Fred Nyhout has developed the argument here, as well as elsewhere, that the wing patterns form a homology system akin to the vertebrate skeleton. So just as you can identify the ulna bone in different vertebrate groups, you can trace the identity of a pattern element across Lepidopteran groups, and you can use those characters to infer phylogenies of the Lepidoptera. So in this respect, the symmetry system is unlike other patterning phenotypes such as leopard spots, zebra stripes, or human fingerprints, which are products of stochastic patterning processes that are unique to each individual. So you can't trace the identity of a specific zebra stripe across different individuals. It doesn't have character identity. So in that case of the zebra, there is homology at the level of the striped phenotype. So having stripes is a homologous character, but there's no homology at the level of specific stripes any more than there's homology of a ridge of your fingerprint. Okay, so another way of saying this is that the zebra stripe is not an individualized character. That's the language that Fred and some of his students use. So what does that really mean? What kind of character are we talking about there? Here's a more specific example. Here's a butterfly from the genus Heliconius, which is known for colorful wing patterning. How should we decompose the wing coloration patterns into characters? What are the true characters here? Well, to most human observers, it looks like we have some spots of yellow pigment and orange pigment against a black background. So maybe the yellow and the orange spots are the characters. And this is actually what the early investigators thought. But it turns out that developmentally, the yellow and the orange areas form just wherever there's an absence of black pigment. So the black is actually the foreground and the rest is the background in a sense. The black pigment arises from point sources that fuse together. I have a video of this from Fred's website that shows it. More. Black pigment arises from these point sources, and then the rest is just filled in as a kind of byproduct. Okay. Now, this insight derives from developmental genetic studies of pigment synthesis, but also from placing the species within a comparative context of the symmetry system of variation. So in some of the other species of Heliconius, you can clearly see how the black looks like spots of pigment against a colored background. So is that it? The black pattern elements are the real characters, and the colored regions are somehow epiphenomenal or not real? I don't think so. So I think we should say the black pattern elements are developmental characters. They're developmentally individualized via a dedicated gene regulatory module. The yellow patterning elements are not developmental characters. They're not developmentally individualized. They're just a byproduct of individualized characters. They're a little bit like the zebra stripes in that respect. But I don't think we should jump from here to saying they're unreal or epiphenomenal. So the fact that the yellow spots are highly conspicuous might mean that they have a signaling function. And in fact, the coloration pattern in these butterflies are thought to have aposematic signaling functions and effects on sexual selection. So individuals with less vivid colors mate less often. Now, in order for the yellow pattern elements to have these signaling functions, what matters is just how they're perceived by other organisms, whether predators or potential mates. So it doesn't matter if they're produced by presence or absence of a pigment synthesis. So I think we should say that the yellow pattern elements are ecological characters because they play a role in an ecological process, such as predation. Both the black and the yellow patterning elements are ecological characters. I also said the yellow appears to be active in sexual selection. Again, it doesn't matter that it's an absence or byproduct of a developmental process. It matters that other organisms' perception of it has an effect. Of course, there also has to be some underlying variation for there to be a response to selection in that trait. Right? So is the yellow pattern element an evolutionary character? Here I think that evolutionary characters, as I mentioned, they split into two kinds. One kind of evolutionary character has to do with current functioning in evolutionary processes, such as sexual selection, the capacity to participate in the process of natural selection. I think the yellow patterning element is an evolutionary functional character, as is the black patterning element. A different kind of evolutionary character has to do with evolutionary history and phylogeny. So this is basically the concept of homology. Are yellow pattern elements homologous across different groups? Well, this is complicated. I think it depends on your view of homology. So if we take a biological or developmental view of homology, like Gunter's view, it's probably not a homologue. So for one thing, pattern elements are not body parts, as he puts it, characters. They're properties of body parts. And there's no dedicated regulatory network controlling yellow pattern elements. If you take a historical or cladistic view of homology, then I think the answer is maybe. Homologues are synapomorphies in this view. They're shared derived characters definitive of a monophyletic group. And anything that characterizes a group in that way can be such a character, even if it's really the absence of something. So you can have an absence of a trait characterizing a group, and that can be a synapomorphy. But I don't know enough of the system to say whether the identity of yellow spots can really be traced across groups, so I don't know if it's really a good homology. So what about the black pattern elements? These are definitely homologous on a cladistic view, but I don't know whether they would be counted as homologous on a biological or developmental view, because just like the yellow pattern elements, they are not body parts, but are properties of body parts. But it's something we can discuss. So, okay, I hope I haven't lost anyone so far. Is this clear so far? The main point that I want to get at is that there are different kinds of characters that need not always coincide. Okay? Let's look back at this quote here. Yellow pattern elements satisfy part of the characterization, but not all of it. They play a role in certain ecological and evolutionary processes, but they arguably don't have causal coherence or a very well-defined identity. So I'm thinking that having a well-defined identity and causal coherence really have more to do with developmental and evolutionary historical character concepts, which are related to homology, but not so much to the others. But that's something I'm thinking about. Okay, let's get a bit more systematic about these four kinds of characters I'm distinguishing. What are the criteria that we can associate with each kind? Developmental characters. I take a developmental character to be characterized by having a dedicated gene regulatory module and possessing variational independence from other developmental characters in the same organism. And further, that two developmental characters are the same kind of character if they have the same kind of genetic architecture. So it's a bit of a loose characterization. The basic idea is that a developmental notion of characters has to do with the production of variation and not necessarily with what happens to that variation, how it gets sorted by population level processes. And developmental characters are individuated by their variational properties. So I take it that it's not just realized variation, but also about dispositions to vary, variability. So to me, this is a very Devo-Evo notion of characters. I associate it with Gunter's work on homology and some of Ingo's writing on homology as well. So here we can, this nice quote from Ingo, as well as asking what makes two structures in two organisms the same character, homology. We should also ask what makes two structures in one organism different characters. I suggest that what makes two homologs distinct characters is that one homolog can vary independently of another. Now, variational independence is a sort of criterion of individuation, but it's not an operational one, it's not diagnostic. So it's more like an additional condition that some putative character should satisfy. So how do we identify characters that potentially have variational independence? So I want to give some examples of more concrete empirical work in this direction. So earlier I was talking rather vaguely about wing patterning elements, but wing patterns have many elements with multiple colors. So how should you decompose the system into its variational structure? Which are the units that have variational independence here? Okay, let's switch over to moths. It's a bit easier to see. So here you can see there are two basic pattern elements on the dorsal forewing of this moth: spots and bands. So we know that black and white spots, they're produced from the same pathway actually, and they differ in their color due to some threshold effect on the morphogen concentration. So which characters here possess variational independence? Is it visually discrete spots or bands or something else? You can see that there's a lot of intraspecific variation here. Sometimes the white forms a continuous band, and sometimes it's more visually discrete spots. So in a recent paper, Gahn and Nyhout marked these spots, then mapped out their size correlations to one another on 89 individuals. The question is, as one spot gets bigger, like say this spot, what happens to all the other spots?
89 individuals from the same species?
Yes, yeah, of this moth. So the relationships are summarized in this correlation matrix. These are relationships between all the spots. So red is a positive correlation, blue is negative, gray is no correlation.
So this question, so they really have exactly the same spots, the different individuals? I thought there was some variation in how many spots they have and so on.
I think they do have the same number of spots, and they identify them not only by their position relative to one another, but also by, as I mentioned at the beginning, the spots are produced when you have this pigment synthesis that interacts with the vein compartments. So you can actually use the relationship to the vein compartments to kind of track their position. Right. So what you get from this correlation matrix is that the spots covary in groups. The groups are the bands running from anterior to posterior. Like that, okay? So if this one gets bigger, this one is much more likely to also get bigger than that one is.
So one is proximal, and
yes.
the 7 is this one.
Yes. Okay. So the bands have a degree of variational independence here, right? But in fact, the bands, according to their hypothesis, the bands are nothing over and above spots that have fused together. So you might see the bands supervene on the spots. If you do some artificial selection experiment targeting some property of the band, you're really just selecting on spots. So Gahn and I call these bands pseudo-characters and compare them to Gould's example of the human chin, another supposed pseudo-character. So the real developmental characters here, they're saying, are the spots. And when spots...
So what exactly is the argument that makes them dismiss bands as units of variation?
That the argument is just that you only get variation in the bands because of variation in the spots. So they don't vary independently of one another. Now, so there's another group that thinks that spots derive from band. They're bisections of bands, so that the bands are the ancestral state actually, and then it's a modification of that gives you spots. So it tends to leads to this difference.
But that seems to be a little ad hoc, right? Of course, in this case, the bands consist of sets of spots, right? And in that respect, it's trivial that they are somehow related to spots, but it doesn't mean that the bands don't have a coherence in their variation that would justify to recognize them as other than tributary.
Yeah, so I'm kind of reporting what they say. I don't necessarily agree with them. So
what the reasoning is.
Yeah, so I think that this result suggests that if variational independence is a matter of degree, then that higher level unit, as it were, possesses a certain degree of variational independence. Yeah.
Which is what you demonstrate.
Yeah, right.
So, question, so are the spots homologous to eye spots in all the class?
I don't know. I think that
I don't think so.
no, I think that the eye spot is in a certain position, so not these.
These are also in a certain position.
Right, not that, but the eye spots, I think, is a homolog of the neutralids, and those are more.
Yeah.
So they are absolutely prior to the origin of eye spots, evolutionarily.
So they don't, because if they have been homologous to eye spots, then it will be clear that they are independent units, right?
Yeah.
Not necessarily. I mean, if you have morphogenetically sort of this band-like compartments in the developing wing, right? Do you know?
Yeah, I think so. Because the compartments of the wing are not stripes that go across the wing. First of all, there is an anterior-posterior boundary, which is pretty strict, and then mostly the compartments are going in proximal distal direction. So I think it's extremely likely that you will have compartments that goes like stripes.
But it's developmentally much later than the compartment that you're talking about.
Yeah, it could be
that would be
superimposed
contradicting
on that.
the well-supported fact that the neutralid body plan has this, bands, sutures systems, which are also, orthogonal to these early compartment boundaries. So, I think this is an extended developmental process in which different units arise. And so, I don't think we can know a priori what we see here is actually putting spots into bands. The bands is the developmental compartment.
But you do find one bands of isotopes also.
Right, exactly. That's the point.
And those are clearly not the isotope is the limit. Yes, because you
that's
can,
not how I understand
because you have the variation. You can remove one of them without the others. You can select on one of them passing independently of the others, and you can see that they develop from some separate foci. So in that case, I think it's really not the
Yeah, but it doesn't explain the irregularities of this symmetry systems that are all organized in bands. I think there are certain aspects of the development that we know more about than others, but others is in phenomenological evidence that they are persistent in morphological characteristics. So I mean, I'm just saying that. I'm just saying we cannot assume that we know everything about the development of those things to make such strong statements.
Could this be a case where your framework distinguishing evolutionary characters and developmental characters gains traction? Because of Günter's point is, well, the bands also show variational independence, and I guess variational independence may make them real evolutionary characters of one sort. But then the argument that Frederick Neuhaus seems to run seems to deny their developmental characters, because the kind of bands are somehow sort of developmentally derivative to these spots.
Yeah, so one way of putting it is that bands possess variational independence, a degree of it, from other bands, right, but not from spots. Right. So once you
if you would go and try to select on a single spot, the whole line will react, because you have very high correlations between them. So in evolutionary sense, the response to selection would be of the whole line, and not of the next line. So you have a lack of independence between the lines, not between the spots.
Yeah, that's an interesting point. Yeah. Of course, I mean, it's also imperfect correlations, right? So perhaps you could break it.
Right? But much higher within than across.
Yeah, indeed. Yeah.
So the quantitative synthesis may help to ask, so how strong are these correlations actually? Strong enough to be labeled in red.
Can always be done.
This has a lot more to do with the hypothesis of the authors than with anything from statistics. So when you get things presented like this, you should be very skeptical. So, which I am.
Glad this is not my word.
Yeah, I understand.
This is not quantitative genetics. This is qualitative
phenotype.
Correlation
is a quantitative measure.
Of course, yeah.
And it's also a measure that is quite hard to interpret in this context, because of how much variation is there really, and is there equal amounts of variation in the different correlations doesn't really tell you too much about variation all the time. Yeah? I mean, you can have large, even if you have a high correlation, you can have a lot of evolutionary independence between the traits if there's a lot of variation in the first place.
Yeah.
And there's additional thing to the heat maps, and I don't want to say that they fall for that trick, but sometimes if you don't know how it's done, it's usually the blue is taken as the smallest and the darkest red as the biggest correlation.
Yeah,
And it doesn't matter. And
Yeah,
then it
kind of scale like
scales,
this, yeah.
right? And it doesn't matter whether the differences are extremely small, they're scaled as maximum and minimum. That's how very many heat maps are done automatically.
But so that's true, but it's not a random
Right.
set of colors here, right?
Well,
So
you have
it
the
is actually
one
topological.
on the diagonal, and the blue are negative. So the red goes from 1
We
to
don't
0.
know whether it's negative or over there.
Yes, that's very
It's
good.
written.
Yeah.
So
It's not just.
It's not just minimum, maximum.
So I think so, which means that, for example, the band 7, there are some spots which are even nearly correlated, close to 1. It doesn't prevent. I agree with your criticism of the thing, Thomas, but I think okay, it seems to identify at least band 7, 6. It starts to be a bit weird with 5, because there is also groups that are much more, nearly as much correlated on both sides. But okay, it seems to identify some.
Also, I mean, you had 98 individuals. That's not a lot for those samples. 98 individuals, wasn't it?
89.
89.
How many
I don't know,
rebels?
more than 89. There are a lot more parameters there than you have the device. That's for sure.
Yeah. So
But I mean,
it's
it could
more
be
just
true,
a,
right? I mean, well, if that's a master, it's true.
It could also be a complete artifact. You just have to,
But
to
let's
trust
assume.
that they are
But for the sake of the argument, let's
Sure.
assume it's true. Okay. Okay.
I'm going to put on my philosopher hat and say this is a conceptual point.
Yeah.
Philosopher's helmet.
Yeah, everything deflects off of
We
it.
can discuss that early January.
Okay, so spots and bands. So they're comparing these to the chin example. If you have discrete spots that get big enough, then they fuse into a continuous band. That's the basic idea. So I wonder, I mean, maybe there's certain contexts in which it's useful to talk about the band as the character, and others where if you actually measure all the spots, then you can dispense with the band or something like that. I think that's an interesting question to discuss. So enough about developmental characters. Now let's look at ecological characters. I haven't really thought about this that much. I distinguish this concept because of cases like the yellow pattern element in Heliconius. The basic idea is just that a property of an organism can play a role in an ecological process without being individualized and also without necessarily being a target of natural selection. But that's not really our concern here. So next, evolutionary functional characters. This is just something that's capable of undergoing selection as a unit, right? So why is this distinct from developmental characters? Well, it's because something can undergo selection without being developmentally individualized. So like the yellow pattern element on Heliconius. Or think of Gould's chin example. The chin, according to Gould, is not a developmental character, but if there were sexual selection on chin shape or size, then in effect, I would say there's an evolutionary functional character, which is a certain growth ratio between the two growth fields, the alveolar and mandibular, right? And that's not an individualized character, it seems to me. So there seems to be some important distinction here. Evolutionary functional characters are based on function and not on structure or homology. Now, there seems to be a common attitude among evolutionary biologists who don't necessarily care that much about development, that the real characters of an organism are determined by function ultimately grounded in fitness. So basically take fitness and decompose it into survival and reproduction, and further decompose it into major fitness components. And from there, you can identify the traits that are contributing to that performance. These traits are effectively the phenotypic dimensions along which the population is evolving, right?
It also makes assumptions about existence of independent genetic variants, which then is a statement about its development, right? But I don't think it is that separate from developmental individualization. But I mean, I see it's not a one-to-one, but it is not that independent. It's actually something that, in my knowledge, for me, Rosenberg has pointed out many decades ago. I think it's an important point that the adaptationist program makes assumptions about development, but doesn't say it.
Yeah.
And in this case, it also requires that you have independent genetic variants, but then you don't get under all developmental scenarios.
Yeah, that's a good point. So, three authors I've encountered that think this way. Lee Altenberg, in his contribution to this 2005 book on modularity in the Vienna series, this is what he calls a function network, which is basically a causal graph from genes to fitness components. And characters are causal variables between genes and fitness components, and then you have screening off the farther you go up. So if you have a certain contribution to fitness at this level or whatever, that screens off all the causal differences at the lower level. It's not that important. Another example, this famous paper, or suggests a method for calculating the effective number of dimensions of an organism or species, knowing only the present fitness relative to the optimum and the maximal change in fitness resulting from mutations of various sizes. The details are not really that important here, but the point is that traits or dimensions are being defined from fitness effects of various mutational perturbations. And interestingly, in this context, the effective dimensionality of an organism is proposed as a measure of complexity, because he was talking about the cost of complexity. Then we have another example. Poole, in this paper, makes a similar move. He suggests that characters define the dimensions along which evolution occurs. So here's a quote. I propose that evolutionary characters are functional pathways. Evolutionary characters are free to take on values which are to a large degree independent of those of other evolutionary characters. So here we get back to like developmental notion of variational independence, actually. Identification of these characters would allow a description of a population sufficient both to predict its evolutionary dynamics and eventual equilibrium state. Poole's approach is to decompose fitness into the life history traits that contribute to it. He also proposes this idea that traits can be viewed as principal components of the G matrix, and once we've mapped all the QTLs that underlie these traits, then we understand the real evolutionary characters of the system. Okay, that's just a quick tour of three examples. A few things to note about this evolutionary functional character notion. It has nothing to do with homology and phylogenetic inference, except perhaps by accident. It seems to me that these approaches would have a limited capacity to capture variational properties and variability, and hence evolvability of traits. So things like cryptic genetic variation have no effect on functioning, but they can affect the variational properties of something. Just an idea. And a fairly obvious point, I doubt that you can really effectively decompose the organism into its evolutionary characters while ignoring developmental biology, or simply defining traits as causal variables lying intermediate between genes and fitness. So it's maybe interesting, but of course it's going to be a limited approach. Next character kind, evolutionary historical characters. I like presenting to this audience because everyone knows what I'm talking about and what homology is, so I can just say this is homology. That's it. So
You're optimistic.
So with these different kinds in view, so let's circle back to the introduction and the roles I distinguished at the beginning. So the question was, what kinds of characters should be used for which roles? Right? I am proposing or speculating that you should use developmental characters for these roles. Measuring organismic complexity, sure. Measuring pleiotropy, sure. Distinguishing phylogenetic groups, no. Just describing morphological evolution, sure. Identifying bearers of fitness, no, not necessarily. Maybe not the best notion for that. Identifying, yeah.
One comment to that. So pleiotropy is used in a very different way. So there's pleiotropy that is defined as variation, covariation.
Yeah.
Variation of pleiotropy, and there's pleiotropy that is defined in developmental way.
Yeah. Okay.
So this is the one that you're probably referring to.
Yeah.
But it's not the same thing. So from the population genetic perspective, it's variation, covariation, right?
Okay. All right.
Yeah.
But it still requires that you define characters in a developmental and small way, right? Because it's not interesting if that would be arbitrary characters. It's only interesting if you have
well, but we can generate pleiotropy without having developmental character.
But it's not terribly interesting anymore. Not interesting.
I know, but it's used for both in both contexts. That's
So I think you get an interesting result here that when you can use two or more different kinds of characters for the same role, like complexity or pleiotropy, then you get multiple concepts of that, like multiple notions of complexity, multiple measures of it, and so on.
I'm not sure I understood why you deleted this other so why is it not relevant for measuring rates of evolution and identifying origins, for example?
Why is it not relevant for identifying novelties?
Yeah, for example, maybe I just missed something.
Well, I mean, in the characterization I gave of developmental characters, it's sort of without respect to it's sort of like to history, right? And I'm thinking of novelty as sort of non-homology view of novelty.
Right, but that's complementary to homology, right? It's sort of, you can separate it.
Can you explain what you kind of mean by it matters for describing morphological evolution, but not for identifying novelty?
Or measuring rates of evolution?
Yeah,
or
well, that's
Well, I cross those out mostly because there's another concept that seems to be more appropriate for that. So I guess you could use it, but you know.
So what is the concept then?
Yeah, right. So many of these will overlap. So I mean, when I cross something out, I don't mean like never do this or something, but that it's certain conceptions
I think it's because the developmental and historical come so close to each other in definition.
Yeah.
That it's hard to put this into one or another because they kind of to both.
This clearly would have to be addressed in any actual paper on the topic or a more serious approach, right? What about the question of weighting rival phylogenetic hypotheses? I think yes, it's relevant there. So recall the bands and the spots thing. So in a congruence test, you shouldn't count bands as an additional character over and above the spots, right? I guess it's a fairly obvious point, but that the causal structure of development, the mechanistic structure of development, informs your phylogenetic hypotheses about which kinds of transformations are more likely, right? As a general point, right? Measuring rates of evolution, I think that the evolutionary notions are probably more suitable for that. But again, there's a question of overlap and dissociation. Establishing comparable units of phenotype. Yes, this is what.
Just an idea from the terminology. Maybe what you're actually talking about are not different character concepts, but perspectives on characters, and then you are not forced to make that strong distinction. Right, so you have a developmental perspective on morphological characters, let's say, that has implications for all kinds of, you have roles here, and then there's another one but it's not just a categorical difference as in the character concept, which forces us to accept.
Okay.
Right?
Yeah. So I think there are many ways of expressing it. Like I'm expressing in terms of kinds, which is maybe a bit of a blunt instrument, but I don't think that like a entity in the world, such as a character, must belong to one and only one kind. Right?
Right. So therefore, it's
So
actually different sort of projections
yes, indeed,
on similar objects.
definitely, yeah,
Fully for us.
yeah. That's kind of what I mean. So okay, let's go to more perspectives and roles. So use evolutionary functional characters for these roles. Measuring organismic complexity. Well, sure, especially in the context of selection and theory of selection and cost of complexity stuff. Measuring phylotropy, sure, for the same reason, maybe in the same context. Distinguishing phylogenetic groups, no. Describing morphological evolution, maybe, but maybe developmental perspectives and evolutionary historical perspectives will be a little bit better. Your options there. Identifying the traits that are bearers of fitness, yes, definitely. Identifying novelties, I don't think so. Weighting rival phylogenetic hypotheses, again, no. Measuring rates of evolution, sure. Assigning differential evolvabilities, I feel like this one really has to do with the overlap between developmental perspectives on characters and evolutionary functional perspective on characters, especially if we're talking about the capacity of a system to generate variations that are potentially adaptive. So it's not just about variability, it's about potentially adaptive systems, and we're talking about the production of variation and what happens to that variation at the same time. Then what about establishing comparable units of phenotype? Yes, but I think especially for purposes of studying convergent evolution, that context. So let's talk about evolutionary historical characters or homologs. Measuring organismic complexity, yes. So no, I've said yes 3 times to that, which gives us multiple character measuring phylotropy. I don't know, maybe. Distinguishing phylogenetic groups, absolutely yes. Describing morphological evolution, yes. Identifying bearers of fitness, no. Evolutionary functional characters, I think, will be better for that. Again, the point is that homologs can have different functions and different contributions to fitness. Identifying novelties, yes. See Wagner and Müller. Weighting rival phylogenetic hypotheses, yes. Measuring rates of evolution, maybe, I don't know. And the rest, maybe, I don't know. And the last one, yes. Okay. So you see like a basic method, rough at the moment, but a method for approaching the character problem. And this is a major part, I think, for part of the work for a theory of characters. So what should a theory of characters do? The first thing I've been talking about is to specify general criteria for different kinds of characters that allow us to satisfy the roles of the character concept in biological reasoning. That's the working notion I'm working with. But there's another thing that I think a theory of characters should try to do. This has already come up in our discussion, which is to determine when these character kinds will overlap and when they will dissociate. And that's, I think, a really interesting issue. So this is getting a bit abstract. The idea is that different kinds of characters will often overlap on the same units. Think of traits that are fairly obvious, like the human eye, right? But kinds of characters can also be dissociated. We saw this with the wing pattern elements in butterflies and moths. So I want to just close with 2 examples of work that does something on this question. So there's the line of thought about variational modularity from Wagner-Altenburg 96 and subsequent papers. I think this work predicts a certain kind of overlap between developmental and evolutionary characters, right? So if you look at diagrams like this one, you see a kind of alignment of developmental characters which have a dedicated regulatory architecture, or like low plastry between characters, strong interactions within the character, and then evolutionary functional characters which perform distinct functions, and then possibly also characters viewed as homologs. So there are many reasons to expect this overlap. There's theoretically grounded reasons to expect it will be the case. One of them concerns the connection between developmental modularity and evolvability that you'll all be familiar with. So if developmental characters and evolutionary characters were systematically dissociated, it might be hard to explain how complexity could evolve via gradual stepwise modification. It's a familiar argument. So this line of thought generates a sort of prediction that characters with an important functional role will eventually acquire a dedicated modular genetic architecture. Right? Here's another example of different kind of work that describes dissociation of kinds of characters, work on developmental system drift. So for anyone who's not familiar, developmental system drift occurs when there's changes in the genes or the network underlying the same character in related taxa. So this means, I would put this in my terminology from this talk as developmental characters can get decoupled from evolutionary characters, both functional and historical evolutionary characters. Yeah. Okay. So this is just expressed in, it's kind of complicated, but here's a quick expression of it in a paper by Von Dassau and Monroe. The criteria determining which variant persists have in all likelihood little to do with the preservation of any particular relationship between mechanism and function. And then they continue, there's no inherent stability to the genotype-phenotype map, no one-to-one gene or developmental process to character relationship, yet we confront the widespread existence of stable patterns whose coherence must be embodied causally within a shifting sand of developmental process beneath it. A lot of this derives from the very basic insight that many structures can perform the same function. And so if you have selection on a function and other forces that are causing structures to just produce underlying variation, then over evolutionary time you can get multiple structures for the same function. So there are different models and mechanisms of ESD, but I should probably just finish at this point. So the key point is just that there are some specific evolutionary factors that can be expected to increase ESD, such as DSD accumulates over phylogenetic distance. So if you're talking about characters that are phylogenetically highly distant from one another, the likelihood that they have exactly the same genetic architecture is lower. It's also increased by things like low population sizes, higher rates of gene co-option, other factors. We can talk about this in the discussion if you want. So the point I want to draw from this is that perhaps some of these models can be used to predict dissociations between kinds of characters or overlap between kinds of characters. But also maybe this question of overlap and dissociation is just inherently unpredictable. So we get back to this question discussed after Alan Love's talk about how predictable evolution really is. So I'll end there. Thanks for your attention.
Okay, so do we have any external listeners?
Yeah, well, there was some.
Well, that's fine. I try to moderate the discussion to prevent you from speaking. But I will do further.
What kind of friends?
Yes, so, well, in 5 minutes we could the thing if there is any question. But, yes, Gilbert.
So let's start with the last point. I think it's a too fast conclusion that developmental systems, as it is defined, would undermine, let's say, developmental modularity or whatever you call it, developmental character concept. Because if you look into, because first of all, not all developmental mechanisms are equally important for realizing a distinct developmental character. And the way I read the literature is that most examples of developmental systems drift have to do with the inducing signals that cells receive in order to make a developmental phase decision. And we know that these are developmentally often very redundant because of multiple signals come that are different molecularly, different molecular kinds that make the signals can sort of push the cell into the same direction, and therefore they are replaceable with each other. And also over evolutionary time, we see that in some species the same cell type or whatever gets induced by one signal and then another species by the other. But that doesn't actually lead to a situation that the cell types or structures like the nematode vulva is not identifiable as the same character anymore. Mm-hmm. Right? On the other hand, all the good examples of highly conserved elements of parts of the developmental network are parts of the developmental network that specify, more or less approximately specify the developmental identity of the group of cells. Right? And so all that is sort of saying that only because some aspects of developmental mechanisms change over evolutionary time, not all of them are equally important with respect to the question of this is the same developmental character or not. Right?
Yeah.
We have to ask what role these parts of the developmental network play in the developmental process of making a certain part different from the other.
Yeah.
So you would then call it a state rather than modification rather than
like it's a modification of the developmental pathway, right?
Yes, I think that the work on this is just not very advanced enough to maybe say that much. The main, I wonder what you think of this example. That's the example I was thinking of, where you have